chapter Biotechnology Principles and Processes. One of the important topics in this biotechnology is, or the applications of biotechnology, recombinant DNA technology. Recombinant DNA technology is a technology by which we desire to introduce a desired fragment of DNA into a host cell so that its DNA is newly combined with a new combination. That combination is called recombinant DNA. This technique is also called as genetic engineering. According to the principles of genetic engineering, the genes are manipulated as per the desire. We introduce the genes of interest which are having advantages, and these advantageous genes or fruitful genes are introduced into the host cell to produce the desired effect. This technology or technique involves the cutting DNA into specific fragments using enzymes. Those enzymes are called as restriction endonucleases. These enzymes act as the molecular scissors. They cut the DNA at the proper sites while we do desire to cut them, and joining the fragments with the help of enzyme DNA ligase. Another enzyme, that DNA enzyme, is called a DNA ligase. This ligase helps in the ligating the genes, ligating the cut or fragmented DNA. Using this technology, the fragments of foreign DNA can be inserted into a vector, which may be plasmid or viruses. This genetic engineering or recombinant DNA technology is practiced. For example, this is a gene of interest which we want to introduce into this host cell, but we cannot obtain this gene afresh. We obtain it by the restriction endonucleases and then join this part of the DNA with a transmitting substance. That transmitting substance is a vector. It may be a vector such as a plasmid, or it may be a virus. So this gene of interest is introduced into this vector by restriction endonuclease and by ligase. Later on, this vector or virus is introduced into the host cell with the help of further enzymes that helps to introduce this DNA or gene of interest into the host cell to produce a desired effect. The steps involved in this technology are: firstly, DNA fragments coding for proteins of interest are synthesized chemically or isolated from an organism. The next step is these DNA fragments are inserted in a restriction endonuclease cleavage site of the vector that does not inactivate any gene required for vector's maintenance. This is an important point. The vector, which is a virus or a plasmid, should survive whenever a new gene is introduced. This means the vector has transferred this part of the DNA. That DNA does not deactivate its maintenance. One more step is there, that recombinant DNA molecules are now introduced into a host to replicate. This is the vector is introduced then into the host for the replication. Whenever this DNA is introduced into the host, due to the replication, it forms multiple copies of the desired gene. Those multiple copies are called as clones. Recipient host cells that have acquired the recombinant DNA are selected. Selection pressure is applied to enrich bacteria with a selectable marker. Desired clones are then characterized to ensure that they maintain the true copies of the DNA segments that was originally cloned. This cloning is done in the host cell, where exact the photocopies of this DNA, which DNA has been introduced, are produced. As a result, multiple copies of the DNA are produced, which involves DNA cloning. This is all about the recombinant DNA technology, or what we call as genetic engineering, manipulation of genes according to the interest.